In the previous lecture, we studied about different kinds of biomolecules such as lipids, nucleic acid, carbohydrates, and proteins. So DNA is a nucleic acid, and today we will study how DNA makes its own copy. DNA is a polymer which is made up of monomers of nucleotides. A nucleotide has three main parts. One is a sugar molecule, a phosphate attached to the sugar molecule, and a nitrogen base. These nitrogen bases can be of four different kinds in a DNA molecule: adenine, cytosine, thymine, or guanine. Polymerization of these nucleotides leads to the formation of RNA or DNA. The main difference between RNA and DNA is in DNA you have thymine, in uracil is present in RNA. Further, DNA is a double-stranded structure and RNA is a single-stranded structure. So this will be the molecule of a DNA with two strands running anti-parallel to each other. Also, there will be hydrogen bonds between these base pairs. There will be a triple bond between adenine and thymine and a double bond between guanine and cytosine. For replication to occur, first the two complementary strands of DNA have to separate. Then the individual nucleotides come and build a complementary strand with the two separated strands. So what is replication? The DNA replication is the process by which a double-stranded DNA molecule makes a copy of its own, producing two identical copies of DNA. This process is essential for cell division because it ensures that same genetic material is passed on to the two daughter cells. So DNA synthesis occurs right before the start of the cell division. So the cell division starts here in the mitotic phase. Before that, it's important that the complete DNA replication has occurred and there are no errors in the DNA formed. The individual copies of DNA will then be passed on to two different daughter cells. If there is any defect in the DNA, there will be a possibility of formation of cancer cells. Let's have a look at the features of DNA replication. If this is your DNA strand, how do you think the two copies of the new DNA strand look like? Will it be that both the strands of two DNA which came from the parent molecule remain together and two new strands make a separate DNA strand? Or one copy of parent strand each goes into the new DNA and a new DNA strand is built along with the parental strand? Or you think that there is breakage in the DNA strand and then new DNA is only filling the gaps created by these breakages? So after experimentation, scientists has found that DNA replicates by method number 2, which is called the semi-conservative method of replication. It's called semi-conservative because one strand comes from the parent and one strand is new. DNA replication has three main steps: initiation of replication, elongation of DNA strand, and termination of replication. DNA replication originates or starts at specific sites called the origin of replication sites. The DNA replication progresses in both the directions around the replication site. So let's say this is the origin of replication. The DNA replication will occur towards both the sites. There are some key enzymes involved at the initiation step. First is helicase. Helicase unwinds the two strands of DNA. This unwinding lets the building of new synthesizing strand of DNA. Second is single strand binding protein. This protein stabilizes the separated DNA strand and prevents them from rebinding with each other. Another important enzyme is the primase. Primase enzyme builds a short strand of RNA at the beginning of DNA replication. Once this short strand of RNA is built, then DNA polymerase can come and start bringing nucleotides for synthesis of DNA strand. The DNA replication occurs simultaneously on the two strands, this leading strand and lagging strand. But one strand is formed continuously because the DNA strand keeps opening and opening. That's why the DNA replication occurs constantly. But the other strand is building in short bits because it has to wait for the strand to open. So a short bits of DNA are synthesized. These short strands of DNA synthesizing in smaller bits are called the Okazaki fragments. Next step is elongation. Once the DNA replication has initiated, now the nucleotides have to come and keep on building the DNA strand. This polymerization occurs by the action of DNA polymerase, which brings in nucleotides and lead to the formation of new DNA strand. As I have already mentioned, there is one leading strand which is built continuously and the other is a lagging strand which is building in shorter bits as the DNA strand opens. Once the DNA replication is completed on the leading strand, the lagging strand has smaller bits of Okazaki fragments. These Okazaki fragments are then ligated or joined with each other with the action of enzyme called DNA ligase. As already mentioned, it is very important that exact same copy of DNA is replicated. If there are any errors, it can lead to severe disorders such as cancer. So, if there are any errors, the proofreading is an essential step. So, DNA polymerase can correct errors by removing the incorrect nucleotides and then bringing the correct nucleotides at specific locations wherever there are errors. There are other kinds of enzymes such as nucleases involved in the mechanism of repair of mismatched nucleotides. These nucleases can be exonucleases, which cuts at the end of the strand, or endonucleases, which cuts somewhere in the middle of the strand. After the successful completion of elongation of DNA strand and proofreading, now the DNA replication has to stop, and the DNA replication is occurring in both the directions and they stop wherever these two replicating strands meet. That is the termination site for termination of DNA replication. The ends of chromosomes are very tightly wound. These are called telomeres, and for opening these telomeres you require the enzyme called telomerase, which then facilitates the replication in these areas. With each cell division, the telomeres keep shortening, and the shortening of these telomeres actually leads to the aging. Why that happens? With each replication cycle, as the telomere shortens, the probability of correct DNA replication reduces. So as you age, there are more chances of having defect in your DNA replication, which leads to problematic proteins in your body, and these accumulation of problematic proteins leads to aging. Now let's have a quick review of the important enzymes involved in DNA replication. We talked about helicase, which opens the two strands and unwinds them. Then is primase, which builds a small fragment of RNA primer. Once the RNA primer is built, then DNA polymerase can come and polymerize the nucleotides to synthesize the DNA strand. After the completion of replication or elongation of these strands, the smaller Okazaki fragments are joined together with the help of DNA ligase. And enzymes like topoisomerase or single strand binding protein stabilizes the unwinding and prevents the two strands of DNA from winding again during the process of replication. The main differences between the replication process in prokaryotes and eukaryotes are: the prokaryotes have a circular DNA. They have only one origin site. The DNA replication continues from both the sides, but only starting at one replication site. This process is very fast, and in a circular motion they are able to complete their replication very fast. Eukaryotes have linear DNA. That's why they have several origin sites. The replication is generally slower, and this method is much more complex with the involvement of several different kinds of enzymes and proteins. As mentioned previously, it's very important to perform the proofreading of replicated DNA. It's important to get rid of any kind of mistakes in the base pairing. But if there are still errors in the replicated DNA, it leads to mutations or changes in the DNA makeup. What can be the causes of such mutations? These mutations in the strands of DNA can be caused by environmental factors such as UV rays or certain chemicals, or mistakes in the DNA replication. So there will be a mismatch. For example, a mismatch you can see here where G is sitting with T, or there can be breaks in the DNA fragment. These leads to accumulation of genetic defects leading to genetic diseases and cancer. So to summarize, DNA replication is a highly regulated and accurate process. It is essential for maintaining genetic stability or genetic inheritance of an organism, and it's an important step right before the cell division. Any kind of errors in replication, although they are rare, but they can have significant biological impact, can cause genetic diseases and also cancer.